Deep inside your tissues right now, billions of damaged cells are actively accelerating the biological aging process. Scientists call them senescent cells, but in the longevity community, they are widely known as zombie cells. These worn-out cells refuse to die, lingering indefinitely while secreting a toxic cocktail of inflammatory signals that damage surrounding healthy tissue. For decades, researchers believed cellular decay was an inevitable, irreversible consequence of growing older. However, groundbreaking research from leading longevity institutes has uncovered a powerful natural plant molecule capable of hunting down and destroying these zombie cells. That compound is Fisetin, a naturally occurring dietary flavonoid found in strawberries, apples, and cucumbers. Today, we explore the science of Fisetin, how Senolytics target cellular senescence, and what current research reveals about natural cell renewal. To understand how Fisetin works, we must first examine what happens inside a cell when it turns senescent. Every healthy cell in the human body is governed by a strict biological clock known as the Hayflick limit. In young tissues, human somatic cells divide a finite number of times before their telomeres shorten and DNA damage accumulates. Under normal circumstances, when a cell suffers unrepairable genetic damage, its internal safety mechanisms trigger apoptosis, or programmed cell death. Specialized immune cells, such as macrophages and natural killer cells, then arrive to clear away the dead cellular debris. However, as we age, cumulative oxidative stress, environmental toxins, and metabolic waste cause certain cells to evade apoptosis. Instead of self-destructing, these cells enter a state of permanent cell cycle arrest, officially becoming senescent. Senescent cells remain metabolically active, but they no longer divide, perform their normal organ functions, or respond to natural apoptosis signals. Think of them as malfunctioning factory workers who refuse to leave the assembly line while actively sabotaging nearby equipment. Over time, as senescent cells accumulate, the body's regenerative capacity declines, leading to organ stiffness and tissue dysfunction. If senescent cells simply sat quietly in tissue, they might not pose a major threat to long-term health. Unfortunately, zombie cells are far from silent bystanders. Senescent cells synthesize and continuously secrete a destructive biochemical mixture known as the senescence-associated secretory phenotype, or SASP. The SASP cocktail consists of pro-inflammatory cytokines, chemokines, extracellular matrix-degrading enzymes, and reactive oxygen species. When released into surrounding tissue, SASP molecules degrade healthy structural collagen, impair stem cell function, and induce chronic low-grade systemic inflammation. Scientists often refer to this persistent, age-related systemic inflammation as inflammaging. Even worse, SASP signals exert a contagious effect on neighboring healthy cells, forcing them into senescence as well. A small cluster of zombie cells in a joint, organ, or blood vessel can gradually infect surrounding tissue, spreading dysfunction throughout the body. Accumulation of senescent cells is directly linked to joint degeneration, arterial stiffness, skin aging, cognitive decline, and metabolic dysfunction. Researchers now consider senescent cell burden to be one of the foundational hallmarks of biological aging. For years, medical researchers sought chemical interventions that could selectively destroy senescent cells while leaving healthy, functional cells untouched. This new class of therapeutic compounds was named Senolytics, derived from the Latin words for old and destroying. In 2018, a landmark study published by researchers at the Mayo Clinic and the University of Minnesota screened a vast library of natural compounds for senolytic potency. Researchers tested flavonoids including quercetin, luteolin, curcumin, epigallocatechin gallate, and Fisetin across human cellular models and aged animal models. Out of all tested natural molecules, Fisetin demonstrated the highest selectivity and potency as a natural senolytic agent. Chemically, Fisetin is a polyphenol belonging to the flavonoid class of plant secondary metabolites. Plants synthesize Fisetin as a protective defense mechanism against solar ultraviolet radiation, fungal pathogens, and environmental stressors. In human biology, Fisetin possesses a unique molecular structure that allows it to cross cell membranes and modulate key intracellular signaling pathways. This high lipophilicity enables Fisetin to distribute effectively into tissues where senescent cells accumulate. How exactly does Fisetin distinguish a damaged senescent cell from a healthy, functional cell? The secret lies in senescent cell anti-apoptotic pathways, known as SASP. Senescent cells survive despite severe internal damage because they upregulate pro-survival protein networks like BCL2, BCLXL, and PI3K ACT. These pro-survival networks act like molecular shields, blocking the cell's internal suicide machinery. Fisetin functions as a targeted multi-pathway inhibitor, selectively disabling the pro-survival shields unique to senescent cells. By temporarily suppressing BCL2 and PI3K signaling in zombie cells, Fisetin re-enables their natural apoptotic machinery. Once the pro-survival shield is dropped, the senescent cell collapses from its own accumulated internal damage and undergoes programmed self-destruction. Crucially, healthy young cells do not rely on these elevated SASP survival networks, meaning Fisetin leaves normal, dividing cells completely unharmed. Once the zombie cells self-destruct, immune macrophages sweep into the tissue, clearing away the debris and creating space for native stem cells to regenerate fresh tissue. This targeted clearance resets the local tissue microenvironment, allowing healthy cells to thrive. The systemic impact of clearing senescent cells with Fisetin extends across multiple organ systems. In preclinical animal studies led by Dr. Laura Niedernhofer, administering Fisetin to aged mice significantly reduced senescent cell markers across fat tissue, liver, spleen, and kidney. The treated animals demonstrated improved physical endurance, enhanced grip strength, and a notable extension in both median and maximum remaining lifespan. Beyond cellular clearance, Fisetin activates AMPK, the master cellular energy sensor that promotes autophagy and mitochondrial biogenesis. Fisetin also downregulates the mTOR pathway, mimicking the beneficial cellular maintenance effects of caloric restriction. In brain tissue, Fisetin crosses the blood-brain barrier, protecting neurons from oxidative stress, reducing microglial neuroinflammation, and supporting synaptic plasticity. In cardiovascular biology, Fisetin helps preserve endothelial function, reducing vascular stiffness and suppressing inflammatory foam cell formation in arteries. Skin health also benefits, as clearing senescent dermal fibroblasts restores collagen synthesis and improves skin elasticity. Furthermore, Fisetin enhances endogenous glutathione production, reinforcing the body's primary antioxidant defense system. Given these remarkable scientific findings, where can you find Fisetin in nature? The single richest dietary source of Fisetin is the strawberry, followed by apples, persimmons, grapes, onions, and cucumbers. However, dietary concentrations in fresh fruit are relatively modest, with fresh strawberries containing roughly 160 micrograms of Fisetin per gram. To match the therapeutic dosages evaluated in clinical senolytic trials, an individual would need to consume several pounds of fresh strawberries daily. Consequently, clinical research focuses on concentrated Fisetin extracts derived from natural botanical sources like Rus succedanea. Promising clinical trials, including human pilot studies at the Mayo Clinic, utilize intermittent hit-and-run dosing protocols. Because senescent cells accumulate slowly over months, senolytic therapy does not require daily administration. Instead, high-dose Fisetin is taken for 2 consecutive days once a month. This hit-and-run strategy minimizes potential side effects while allowing the body ample time to clear cellular debris and regenerate healthy tissue. Researchers emphasize that consistency in lifestyle factors like sleep, exercise, and balanced nutrition remains essential alongside any senolytic approach. The scientific understanding of cellular senescence and natural senolytics represents one of the most exciting frontiers in modern preventive health. By targeting the root biological drivers of cellular decay, molecules like Fisetin offer compelling insights into how we may preserve tissue vitality into advanced age. Please remember that while Fisetin research is highly promising, high-dose supplementation should be approached with care. This video is intended strictly for general educational purposes and does not constitute formal medical advice, diagnosis, or treatment recommendations. Always consult a licensed physician or qualified healthcare provider before introducing new high-dose supplements or altering your wellness routine. If you enjoyed this evidence-based longevity deep dive, please like this video, share your thoughts in the comments,